Bom dia a todos. Good morning, everyone. Now switching to English. Thank you very much for your presence here for another Scientia meeting online, the last Scientia meeting of 2020. We welcome today researcher Ana Sofia Reboleira from C3C, the Faculty of Sciences of the University of Lisbon. Thank you very much in advance for accepting our invitation. Our talk today is entitled Life in Caves, Records of the Past and Strategic Resources for Science and for the Future of Humanity. To say a few words and present Professor Ana Reboleira, we have also with us Professor Jorge Palmeirim, whom I also thank being present here, and to whom I will now give the floor to say a few words. Thank you. We are not hearing yet, sorry.
These things should be smarter, turning on and turning off the microphone automatically when they know that we need it. Good afternoon. Thank you very much for the presence of all of you. Today's conversation will be led by Ana Sofia Reboleira. Ana Sofia is a recent member of the Department of Animal Biology, where she was hired as an assistant professor, and she is also a recent member of C3C, of our group, Tropical Mediterranean Biodiversity within C3C. Sofia came from the Natural History Museum of Copenhagen in Denmark, and she works mostly on a very original environment in underground habitats. We tend to forget that because we don't usually go underground, we tend to forget that there is that one half of our ecosystem which is under our feet, and it's precisely in that ecosystem that Sofia has been working on for most of her professional life. She has done a lot of work on taxonomy of underground organisms, particularly invertebrates, and she has described many species of new species of fauna from underground environments, from Portugal and from elsewhere throughout the world. She has worked and done field work in many places of the world, and now she's also working on different issues, not just taxonomy, but also on ecological issues, mostly relating to, but not only, underground organisms. So it's certainly going to be a very different trip, an underground trip today, and it's certainly going to be very interesting. Thank you, Sofia, for being willing to talk to us. Whenever you want.
Thank you, George. Good afternoon, everyone. I will talk now about a subject that I like very much, which is life in caves. So caves are connected to humans since humans are humans. So there are records of our presence in caves. And when we talk about cave biodiversity, that record is slightly more recent. It has 12,000 years from the Magdalenian period. And there is what we call an illustration, probably the first illustration of a cave animal, which was an orthopteran, that was marked in a bone of an auroch. It was found in a cave in France. Throughout the history of humanity, caves have been linked to humanity. So if we go to the Greek mythology, for example, we can see that Rhea, the mother of the mountains, gives birth to Zeus in a cave located in Crete. Also, the brother of Zeus, Hades, who is the king of the underworld, often represented in caves. But this is not exclusive of the Japanese mythology. If we go to Oriental mythologies like the Japanese one, we can see that Amaterasu was the goddess of the sun. She hides daily in a cave and then appears again. And that's how we have the natural cadence of the day and the night. what we call in science the circadian rhythm that affects all living creatures, at least at surface. Then throughout the Middle Ages, the caves were mostly considered gates from hell. They were guarded by Cerbero, which was this mythological creature with two heads. And caves were thought to be inhabited by fantastic creatures. Those included dragons, as we can see here in a representation of a dragon. This is actually the symbol of the International Society for Subterranean Biology. And all these thoughts were deeply rooted in the religious thought. Then we had to wait until the 17th century to start to see the first organized expeditions. In fact, these expeditions were organized to search for animals, but they were probably not searching for the same animals as we are nowadays, since they were searching for unicorns. Then the beginning of, let's say, the science of cave animals started in the end of the 17th century, more or less by the hand of Slovenian noble. And he thought he found the first evidence that actually dragons inhabited caves. So he thought he found a dragon larvae. We had to wait almost 100 years for the formal scientific description of this animal, which is not more than a cave salamander. So this animal inhabits the Dinaric karst, so the Balkan Peninsula. And well, now we know that this is actually the adult. And it's very interesting because these animals have what we call the neoteny, which means that they preserve juvenile characteristics in their adult stage, like this sternal branchia. The first studies on cave biodiversity appears actually by the hand of a Danish researcher. He was a professor on the Zoological Museum of the University of Copenhagen, where I worked until last August. And he published in 1839 a book about subterranean fauna. This book was firstly published in Danish, which is not, let's say, a very popular language among internationals. But this book was translated to English under the title of Specimen Faunae Subterraneae in 1851. And this was very important. Schut provided the earliest classification of subterranean animals. And this was very, very much important because his ideas about adaptation of species to the environment was fundamental for Charles Darwin's inspiration. Actually, in the chapter 5 on the origin of species, published 8 years later, Charles Darwin cited extensively Schut's work as a strong evidence of natural selection, what we call natural selection. Well, my research area is mainly the biospeleology. So this means the biology of caves. When we think about caves and about working in subterranean ecosystems, normally we think about beautiful places, large spaces in the underground, full of beautiful crystals. But the reality is that most of these places are very narrow, very muddy, and especially very wet. This is one of the most important features of caves and subterranean ecosystems, because all the water that infiltrates at the surface starts circulating in the underground, and that's where we have stored 97% of the global resources of fresh water that is immediately available for human consumption. So we must take care of this water, as this is a strategical resource for the future. When we compare subterranean and surface ecosystems, some ecological features change dramatically. First, we have no light. There's absolutely no light in the underground. So when we go to a cave, everything is pitch black. And unless we have a light ourselves, we cannot see in the dark. Then at surface, we have a very pronounced air temperature variation, while in the underground, the changes are very subtle. We have measurements of temperature variation every hour for caves in Portugal, and the variation is smaller than 1 degree throughout the entire year. Then, having no light, we have no plants, or at least we have no superficial part of plants from which all life depends on, directly or indirectly. So when we go deep into the underground, the organic matter amount reduces considerably. And when we talk about the terrestrial compartment of caves, the humidity is remarkably high. So this will have deep changes. This will entail deep changes into the animals that live in the underground, into the structure of their food webs, and many of the ecological processes occurring below the ground. So when we think about caves and this type of caves that we are more used to, the karst caves, so caves on limestone, this one that have this very typical stalactites and stalagmites, they occur in limestone areas, so karstifiable rocks. And when we look at the world distribution of karstifiable rocks, we can see that they account for around 50% of the surface of our planet. But caves are not only in limestone, caves are also found in volcanic territories. And in these places, they are not the result of the action of the water dissolving the rock, but they are the result of fire, so of melted rock that forms lava tubes. So this is how a lava tube looks like. This is in Tenerife, Cueva del Viento, and this is my PhD co-supervisor. So in ecosystem, this subterranean ecosystems are very important to us and to nature because they provide what we call ecosystem services. So they act at the structural level, so they store water, they buffer floods and droughts. They are also an habitat refuge of animals that provide us water purification, among other things, and they are also very important culturally, spiritually, and for recreative purposes. However, everything that we do with surface by gravity will go deep into the underground, and it will have severe consequences on the biodiversity that provides us these ecosystem services. But when we look at the legislation and the protection of the specific ecosystems and the species that live in the underground, and if we have a look at the EU Water Framework Directive, for instance, or the Groundwater Directive, we can see that they aim for the good physico-chemical status of the water, but they completely neglect their biodiversity. And if we look at the Habitats Directive, also, there are very few species of cave-adapted animals listed. So the main challenge of my latest research projects are to evaluate the impact of anthropogenic activities in subterranean biodiversity. This is particularly difficult because below the ground, we have species that are slightly different from what we have at surface. So just to agree on some terminology that I will be using from now on, we have superficial habitats that we also can call epigean habitats. in opposition to hypogean habitats, which are called subterranean. Then within the subterranean, we have the edaphic habitats that correspond to the soil layers. And then it is after the last layer of the soil, the sea layer, that we actually start to have what we call the subterranean, real subterranean habitats. So these include terrestrial and caves, and also aquatic in groundwater. So animals that live in all these ecosystems, they're going to be different. The ones that live deeply in the underground and mostly in large spaces like caves, they have suffered convergent evolution. So the severe ecological features like the lack of light made them lose their eyes, because they don't need eyes to see in the dark, made them lose their pigments. And on the other hand, their antennae become more elongated, and they develop several sensorial organs so that they can feel what is going on in the outside. Then cave animals are typically represented by small populations because we have low food in these ecosystems, and they are very locally adapted. So we have species with very short range distributions. And this will be a serious problem when they face threats, because we cannot go elsewhere and pick new populations to reinstall in the same place. Apart from that, when we want to work with these animals in the lab, they have very slow metabolism. So the studies take very long. They are also very difficult to breed and to maintain in the lab, and they have very reduced fertility. So all this combined represents a big, big challenge to work with. So one of our first questions was, are subterranean species more sensitive than surface species? And this, well, given all these considerations and characteristics, we would expect them to be more sensitive. But looks like it's not really right. So we started with an eco-evolutionary approach in which we modeled comparative toxicity responses. And for that, we selected two different ecotypes. So a surface one and a cave one. And then we selected across a calibrated phylogeny, we selected pairs of close related surface and subterranean taxa. And we did this across different genera, different lineages within the same genera, and even different populations within the same species. Just as a curiosity, these two animals that you see in here, they belong to the same species. This is Azellus aquaticus, in which this lives in a river outside in Slovenia, and this lives in a cave in the same river, but below the ground. So it's really spectacular how they can change so much just by living in different ecosystems. So we had a good model, and we decided to go out in the field and start working to answer our research question. The problem is that most of these species do not survive out of their caves. So how could we run experiments that are standardized so that we can compare responses if we can't bring the animals to the lab? Well, we brought the lab into the cave. So we packed the lab and we run all experiments in situ, so both in the cave and also in rivers with the surface species. This was not so straightforward because we also had to develop a protocol so that we could run this experiment and have faithful results. And then we just had to pack and fly because all these places are located in different parts of Europe. So traveling light as usual and spending lots of days in the underground running experiments and also at surface. So I will not present the results yet because they are part of a PhD thesis of my student Andrea Castaño Sanchez, which was just submitted last month. And this is also research funded by a Willem Young investigated that I was given in 2017. But the results will soon be published. So now we are in Portugal, or well, right now we are more or less somewhere on the internet, but physically we are all in Portugal. So I'm going to be talking a little bit about my research and what lives in caves of Portugal. So Portugal has two main karst areas. So we have two main areas where cave animals are distributed. So these are located in the west of the country and includes the biggest, the largest karst massive of Portugal, the Estremanho. And it's also located in southern Portugal in the Algarve. Then we have some smaller and more older areas. So these are mostly Jurassic limestone. And these ones in the interior of the country are more old rocks. So these are not really limestone. Most of them are marbles and similar rocks. So the story of biospeleology in Portugal started during the last century. So we have the description of the first cave animal in 1930, and then we have had the major input during the decade, during the 40s, by what is considered the pioneer of subterranean biology in Portugal, which was Professor António de Barros Machado. Then we have had several decades with several intents, but not so many results. And when we got into the new millennium, this number changed dramatically as a result of the efforts of many persons combined. Well, this was actually featured in Science. As with the result of this, we were able to get into the list of the world's hotspots for cave biodiversity. So the hotspot that we have currently in Portugal is a cave located in the Algarve in the south of the country. And that cave becomes richer and richer the more we work in there, which is normally the pattern. Richest areas get even more rich. So one of the species that we described from that cave in 2010, we named it the giant pseudoscorpion of caves of the Algarve. And that is because when we compare the size of this pseudoscorpion, we can see here a normal size pseudoscorpion. It's around 3 millimeters, more or less. Then here we can see another cave pseudoscorpion, which is slightly bigger. We can see some characteristics. It has no eyes. It has elongated appendages. And what we found in the Algarve was this. So this is why we call it the giant pseudoscorpion from caves of Portugal. This animal is very spectacular in appearance, and that got him into the list of the 100 most amazing new species of our country. Well, cave animals are really cool. I really like them, but I think we should go deeper into this subject on what is the contribution of the study of subterranean ecosystems for science. So first of all, they are very important in the biogeographic history of animals. Most of them are what we call living fossils. And this is what Charles Darwin called the wrecks of ancient life. If we look at the pseudoscorpion that I just showed you in the previous slides, and if we look at the world distribution of the family to which he belongs to, which is the family Bucicidae, we can see that we have the records here in the Iberian Peninsula, and then all the other representatives are at the other side of the Atlantic. And this means that these animals are so old, but so old in our territory that they were already in here before the continental split. So they can provide us very interesting and historical biogeographic information. On the other hand, cave animals are known to be models for ecology and evolution. And that is because below the ground, we have simplified ecological processes. So we reduce considerably the amount of variables, and we can more easily sometimes make some more accurate observations about biological processes when we use caves, in a similar way to what we do when we use islands for research. So one of these cases is a work we published last year, and which we study a case of hyperparasitism. So in caves, we have bats, as Professor Jorge Palmeirim has been working on for more than 40 years, probably. And on the top of those bats, we have some very specialized parasites. So these parasites are actually flies. They do not look like, because they lost their wings, and they have their mouth adapted to feed on blood from the bats. And they live in the fur of the bats. So this is a parasite. And on the top of those flies, we have some fungi that grows exclusively on top of that fly, and that makes the fungus a hyperparasite. So in caves provide a very simplified model, because these bats, they hibernate in the caves, so they are very much in there. So we can study with some security, some issues about this that we cannot do when we study them on bats from the outside. Then also for medicine, caves and life in caves is very important, as caves are also a source of new antibiotics. So microbiomes from caves, they produce several new substances and bioproducts that have been used on their potential for drug discovery and for their use in medicine, and mostly on antibiotics, the antibiotic effect of them. Then caves also contribute enormously to the biogeochemical cycles in our planet. So we tend to look at the cycles as something that happens at the surface, but it is not. All the nutrients go down and they are recycled, and then somehow they come out to the surface again. And this is particularly relevant in the carbon cycle, in which a considerable part of the decomposition in our planet occurs below the ground. And then this carbon is returned to surface by respiration and by the openings of the underground to the atmosphere. But again, finishing as I start, I deeply believe that one of the most important features of subterranean ecosystems are the groundwater resources for the world. This is important for humans, for us to drink water, but this is important for all life on Earth. Without these water deposits, we will not survive. So, how do we study subterranean hidden ecosystems? So if we are talking about very deep caves, as the world's deepest caves located in Western Caucasus, this will include expeditions that last more than 2 weeks. We have to carry all the equipment up to the mountain, and we have to feed around 30 persons during 1 month. So this is the equivalent to 5 to 6 tons of equipment that we have to carry up to the mountains. Then we have to hang on ropes because most caves are not horizontal. And then deep caves, we cannot go down and up in the same day because they are too large. So what we do is that we actually set up camps in the underground. So especially in what we call the alpine caves, the life conditions are, let's say, miserable. There is 100% humidity, there is water everywhere, temperatures are extremely low, and humidity is 100%. So these are really, really, really tough conditions to work. And these base camps that we establish give us, well, the minimum comfort conditions to feed and to rest and to keep working. Then, apart from surviving in this environment during the expedition, we also have to sample. Cave animals are rare. They are difficult to find. So this is a work that takes a lot of time. And a lot of time in freezing conditions is something that it's really difficult. But this sometimes pays off very nicely. In 2012, we described the world's deepest subterranean community. So the first living below 2,000 meters from the surface. So we didn't find one animal, we found a complete subterranean community living in the underground. And that included the deepest terrestrial animal ever, which was this springtail, illustrated in here. And that soon got to know what the meaning of a viral thing on internet is. But not all the work we do on subterranean ecosystems includes going deep into the underground. Sometimes we can also work at surface. As this case, this is field work we were conducting with our colleague Grant Oos in Australia. So this is an aquifer in sandstone. And we were actually working from pipes. So we were extracting water with an inertia pump to study the animals that were living inside. And then with these animals, we tested the effect of salinity, because salinization of groundwater is a very serious threat to both to water quality and to biodiversity, and also to estimate the effects of temperature in these ecosystems. According to our predictions that you can see in this paper, by 2017, a considerable percentage of these animals will be threatened by global warming. Then apart from that, we also have a platform for society and science, for outreach, which is called Troglobiario. Troglobiario is our cave lab. It can be found in the largest massif of Portugal, in the natural park of Serra da Candeeiros. This is the largest subterranean chamber of Portugal. We have an elevator until minus 33 meters. And then we have inox structure, and we have terrariums and aquariums in situ, so inside the cave. Inside these terrariums and aquariums, we have cave animals that society can observe. You need to book in advance before you can go there. So this serves both society, that allows the observation in situ of cave fauna, which is something that is very rare. And then for science, to us, because it provides us a subterranean lab where we can run a wide array of experiments with these cave animals in their natural environment. So here are two students of mine renovating the specimens in the aquarium and running some experiments. And now to finish, I'm gonna show you a little bit of who lives in caves of Portugal. So here's the nymph of the pseudoscorpion I mentioned before. So very, very large predator walking in the caves of the Algarve. Here we have a cave spider. This was the first cave spider described in Portugal. It lives in the Estremanho Massif, and it carries a massive eggs. Then we have running spiders also, this one from the Algarve, which is hunting and running in the floor of caves in Algarve. We have opiliones, very, very small animals. And we have centipedes, so new species of centipede that doesn't exist yet. We have cave millipedes. Now we have several species of cave millipedes, but the first one was described in 2013. So not so long ago. This was described 2 years ago from the cave where we have our subterranean lab. And when we look at these animals in the scanning electron microscope, we can see lots of details. As the lack of eyes, the eyes of millipedes should be in here. So it's a completely blind species. We can look at the tip of the antenna to see their sensilla. And we can also look at the tip of their claws, which is very typical of cave animals. So this is the accessory claw, which is very small in cave animals. And for instance, in millipedes that climb trees, this one is very large. Then we also have aquatic animals, Proazelus lusitanicus, also a species you can observe in the cave lab. We also have terrestrial isopods. Well, and we can, as you can see, they roll up. And we have Diplura, sister group to insects, 2 very large antennae, 2 very large cerci at the end of the body, so that the animal can explore the huge area while walking. Here's the face, the frontal view of the head of the animal, and the detail of the cuticular microsculpture of the animal, and even more detail. So you can see the scale, this is 1 micron. Then we have Thysanurans. So Zygentoma, this is the cousin of the silverfish that you have in your toilets, that you see mostly during the night. Very large species. This is the largest terrestrial insect, cave insect of Europe. And we also have some animals feeding on the roots of plants that hang from the ceiling. Next year will be the International Year of Caves and Karst. And we will have for the first time the International Cave Animal of the Year, which next year will be the cave beetles. So we have several cave beetles in caves of Portugal. We have several species of the genus Trecus. We have 1 species in the Algarve. And you can see here in the detail of the scanning electron microscope that when we think that we are looking at 1 animal, actually we are looking at 1 animal, several fungi that are growing on the electron, and another animal, an acari, that is living also in the animal. So we never have 1 single animal. We have a community of living things living in the same place. Then we have staphylinid beetles as well. And last year we described the first groundwater beetle from Portugal. And now to finish, we also have bats. I do not study bats directly, but I think it's important to talk about bats, especially, and the importance of bats, especially this year, where because of this COVID-19 situation and the link to bats, they have become even less popular than they were. Bats are very important for ecosystems. They control insect plagues, and they are fabulous. And if they are the source of the problem, they are probably also the source of the problem. To solve the problem. Because if they can handle this coronavirus and they are mammals, so will we in the future, I hope. Apart from that, they have very nice lips. Don't you agree? Thank you very much. This that I present in here is the result of the work of many people. Unfortunately, they do not fit in here, so you can read the acknowledgement list on my webpage. And thank you very much also to the funding bodies that have supported my research throughout the years. Thank you.
Thank you. Thank you very much, Anna, for taking us in this incredible journey and learning a little bit more about what it's the life that is hidden to our eyes. I was very curious. I have some questions myself, but I will go first to the questions that we have on the YouTube chat. And I invite everyone that is following the session to also leave your questions on the YouTube chat. We now have some time for questions. I will start. We have 2 questions already. I will say first one, and then I'll give you time to answer, because I think we have time to develop a little bit on each of the questions and not to confuse each one of them. So first question by Margarida Matos is, how can you tell whether the differences between cave populations and non-cave populations are due to plastic responses versus genetic evolutionary responses. Could you use a common garden design?
We have molecular phylogenies for this, and transcriptomic and genomic data. So that's how they are, species versus populations.
Okay, thank you. And a second question by Margarita Matos as well. She asks, are you able to breed populations in the Subterranean lab? For example, that might allow transplant experiments to test for evolutionary differences between subterranean versus surface populations.
Well, it depends on the animal. There are some animals which we succeeded in breeding on, and some others, which is the vast majority, that we don't. And then we have populations are very small, the metabolism is very reduced, and the fertility is also very low. So these animals, they are strategy K. So that means that they put less eggs, and but the eggs are bigger, so they have more nutritive substances. So we can do a lot of things, but the things of working especially with live animals, which is what I've been trying to do and doing over the last decade, is extremely difficult. And we are always very limited in the number of specimens that we work with. So for instance, this when we studied this comparative toxicological responses, we needed 60 animals, and getting 60 animals of cave representatives and making sure that they are all alive and that they get to the end of the experiment alive so we can validate, it's extremely difficult. So that's also a serious limitation. It's not impossible, nothing is impossible, but working in the underground normally takes way more time than running experiments with, Drosophila or some other Daphnia, some other model organism that we can obtain fast generations in a short period of time more easily.
Thank you. We have another question, which I think is related to what you were already saying, by Ivo Cello. He congratulates you, nice talk, and he asks, is it known where the difficulty in bringing cave populations to the lab comes from? Is it micronutrients, physical conditions, temperature and humidity, etc.?
Yeah. So, this was actually when we started this experiment, we had the problem with the first species. So, and there were previous studies on that. So we used that also to decide on what to do. So for instance, this species that we're working with, which is a species of an azalid from the Jura karst in France, it has a very, very small thermal range. So it survives, if I remember properly, the habitat is 10 degrees and it survives up to 12 and down to 7 degrees or something like that. So it is very, very difficult to transport them to the lab, which we were working in a lab of our colleagues in the CNR in the University of Lyon. So going from the Jura with the animals to the lab and not exceeding or not going down this thermal range was very difficult. So temperature is one problem, but now we are running experiments with cave animals from Portugal, and so far they are extremely tolerant. So we don't really know. The thing is that most of them do not reproduce, and I don't know if they do not reproduce at all in lab conditions, or if their life cycles are just so long that we do not have them enough time to reproduce them. However, there are some species, especially here from Portugal, that we have in stock and cultures in the lab, and that have been reproducing once a year, which is very good, producing an offspring of 14 juveniles. So that's the best we can do.
Okay, thank you. We now have one comment and one question. I will start by the comment and then introduce the question. So the comment is by Margarida Matos concerning one of the previous questions. She mentions that the question was not whether they were genetically different in general, but whether the phenotypic pattern, example, lack of pigmentation, is genetic. So this is the question.
Yeah, I can answer that. Well, I do not work on that, right? So let's start from the beginning. I do not work on gene expression, phenotypes, and something like that. But for instance, we have collected, there is this Procambarus clarki, this invasive, how do you call that? Shrimp crab, what's the name in English? Well, Procambarus clarki, it's a crustacean. The red crayfish. The red crayfish gets into karst springs and is now found in caves of Portugal and also in Italy, and I'm pretty sure elsewhere, but it was not reported yet. And we're talking about a red gray shrimp. And when we find them in caves, they have huge eyes, but they are completely depigmented. So they have fully developed and pigmented eyes, but their bodies are completely depigmented. As soon as we take them out, they start to be a little bit pinkish when they get sun. So the mechanism that controls the pigmentation, well, it's fast because it blocks, but we know that, right? So when we are in winter, we also do not have the same color as we have when we go to the beach. So something like that. But then the mechanism that controls the eye expression is completely different, and that will probably take longer. But one thing we know is that the phenotype is not time related to the colonization of the underground. So there are other factors like intraspecific competition or other factors that we do not know that play a major role in shaping this, the phenotypes more than time.
Okay, thank you. And now going to the question that I had mentioned from Dimitra Petrocello. I think I'm mentioning correctly. She says, greetings from Greece. So we are reaching Greece. Thank you for the presentation. I didn't really catch why the cave species have no body pigments. Could you explain this more?
Yeah, so body pigmentation is a protection. Pigmentation is a protection from UV light produced by the sun. If they live in an environment where UV light doesn't exist, they do not need to produce pigment. So that's why we have the pigmented animals. The same goes for vision, but as we just recently discussed, genes and all that are different, the ones that control that, right? So the opsin gene and all that.
Okay, thank you. Another question. So I have questions coming up by Yves Shell again. He asks, are caves overall isolated from surface, or is there the possibility of migration, that is migration from other caves or the surface?
Yeah, so caves are isolated to some extent, and they are not isolated to the other extent. So let's see. So they are always connected to surface because by gravity, we always have water infiltrating from the surface. There are another type of caves. This is what we call the epigenic caves. So caves that are formed from the surface down to the underground. But there are other type of caves, which are the hypogenetic caves that are formed in the opposite way. And they are formed by external fluxes of thermal waters that will dissolve the rock until it reaches to the surface, right? So in those cases, they are probably more isolated systems. The thing is, we should look at probably at caves as we look at islands and archipelagos, right? So an animal can always go from one island to the other, but it has water and sea all around. In here, the animal can go probably from one karst massif to the other, because it might find a continuum of some kind of subterranean ecosystem, a shallow one, like an MSS, something like that. It's extremely unlikely. And then, especially in the terrestrial cave animals, humidity is a very limiting factor, because the animals live in 100% humidity, so they lose the mechanism of protection to the section. So when we bring these animals to the surface, if they are in low humidity, they will simply dry. So it's probably more easy for that dispersal, a groundwater animal that can go through the hyporheic zone of rivers, and which there is a continuum, and we can see that on the dispersal of groundwater animals and their ancestors, especially related to the regression of glaciers in Europe. So we can see that they went through the rivers and then adapted locally to different karst areas or other type of aquifers. So.
Thank you. I now ask a question myself, and Professor Spalletti, if you also have any question, please let us know. My question, I don't know if this is a dumb question, but taking into account all the hidden life and all the discoveries in the last decades of new species in Portugal and worldwide concerning cave habitats, is there any estimate of what we don't know yet? Is it possible to have an idea?
Well, there was an estimate back in, I don't remember if it was 2000 or I think it was 92, that we would have around 10,000 species. But that's we have more than 10,000 in Europe for sure already. So yeah. And richest areas like the temperate and tropical areas will give a lot of species for the next generations if we do not destroy the subterranean environment with pollution and another type of destruction. So I mean, I do not know. We don't know anything. We did estimates for Portugal. And we estimated that in the Algarve, we knew between, if I remember properly, between 70% and 91% of all cave animals. This was in 2012. We already exceeded the number of, so we already have more species than the ones we estimated. And we estimated that here in the central Portugal, in the Estremanha Massive, I think it was around 50% of the known species. Okay. And we have not exceeded that, but it's I mean, the Algarve is much richer than our center, so it's...
Okay, thank you. So
can I?
Yeah, please.
Well, two questions. One is if there is if the underground organisms show the same trend of greater richness in the tropical regions than in the temperate zones, and in sequence of that, why do you think that in Algarve we have more species than in the Extremaduran limestones?
That's a very interesting question. The answer is we do not know. So first of all, the data that we have for subterranean adapted species, so this cave adapted species that do not live only in caves, because caves is just a place where we fit, right? So the subterranean ecosystem, it's much more extent. So the vast majority of what we know is here in Europe. If we look at all Africa, for instance, there is very, very, very little work done. In the tropics, so in Europe, we have 100 years, slightly more than 100 years of research. Most of this research was concentrated in the Balkan Peninsula. And tropical areas were started, I mean, they are now like in Brazil, they are studying a lot, but this study has around 20 years. So it's not comparable in terms of results. And hotspots like in South America, they are appearing almost every year. Then we know a little bit more on North American cave fauna as well. And like when we go to China again, which is extremely rich in cave fauna, we also do not know a lot. So we know of some groups like beetles and some more popular invertebrate groups. What we do know from Europe in a paper by Culver et al. From 2006 is that the richest areas along Europe correspond with areas of higher primary production at surface, which makes sense because if we have more food at surface, when it infiltrates, we will have more energy in the caves, and that will be able to maintain a richer community. Now, if we have more species in the tropics or here in the temperate zones, we do not have comparable data for that. But I mean, below the ground, we should follow the same pattern as at surface. So.
And what about Algarve versus Extremadura?
Algarve versus Extremadura. Well, that's a very interesting question. So I don't know. Actually, I don't know. The Algarve has many influences. It's a drier place. Being a drier place, animals tend to go more into the underground. We have way much water here in the Extremadura Massive or Sico, for instance, than water available than we have in Algarve. And that can be a driver for species to go deep into the underground. Then we have proximity with Africa. So, I mean, 5 million years ago, we were connected to Africa. So many lineages were able probably to cross and to reach the Algarve sooner than they reached the center of Portugal. But the real reason, we don't know. Also, the Algarve has a lot of area also. But I mean, the Extremadura Massive also have. So, I mean, all the basic parameters that we could use is species versus area, and they fail on that. So it's probably more related to climatic reasons.
Okay, thank you. Thank you once again. I'm looking, for now, we don't have more questions and we are approaching the end time of this session. So I would propose us to wrap up. And I think we can summarize that there's a lot that you have discovered and the scientific community has discovered. And there's a lot we know that we don't know yet. So there's a lot to explore. So it's a very exciting area. So I became very curious and I'll search for more about this afterwards. So thank you once again for your amazing talk and thank you for accepting our invitation. Thank you also, Professor Palmeirim, for being here with us and joining this session. Thanks, Mariana, who is here, Mariana Marques on the backstage and helping everything go well. And thank you all that have followed this webinar and stay tuned for the next webinars in January. So now we will have a break for holidays. We wish you a Merry Christmas and a better 2021. So thank you very much.